Hello friends, this video on Biomolecules Part 21 is brought to you by examfear.com. No more fear from exam. We will now talk about the next group of biomolecule, that is nucleic acids. So till now we have covered two biomolecules, that is carbohydrate and proteins. So now we will talk about nucleic acids. Let us now talk about nucleic acids. So what are nucleic acids? The name nucleic acid itself sounds quite interesting. Why are they called nucleic acid? It is something related with the nucleus, and that is why the name. Now, talking about nucleic acid, let us talk about something which is very interesting. We see our hereditary traits. We see our traits which are similar to our parents or grandparents. So the inheritance which take place, that is the passing of similar traits from one generation to the next. That happens due to the information contained in something known as the DNA. So what is this DNA actually? You would have often heard this term DNA. While we were talking about cell also, we spoke about DNA, chromosomes, RNA. So what is this DNA, RNA? What are they? They are nothing but biomolecules. They are nothing but nucleic acids. So nucleic acids are concerned with this inheritance, the genetic behavior. So with all these things, nucleic acids are concerned. They are known as nucleic acid because they are present inside the nucleus of the cell. So when you talk about a cell, you have a nucleus inside the cell, and inside the nucleus you have all the genetic material. For example, this is your nucleus. Inside the nucleus, you have thread-like structures called chromatin. This chromatin later condense to form chromosomes, and the chromosomes have genes, and the genes have DNA. So DNA is the molecule which actually contains the genetic information. So it stores the information which it passes from one generation to the next. So this DNA is nothing but a nucleic acid. So when you look at the structure of a nucleic acid, you end up seeing that they are polymers, and they are made up of several nucleotides. Now we will see what are nucleotides. Now when we discuss about nucleic acid, there are two types of nucleic acids which we'll come across. DNA, that is deoxyribonucleic acid. And the next one is RNA, which is ribonucleic acid. So these are the two nucleic acid. DNA, which is deoxyribonucleic acid. RNA, which is ribonucleic acid. So DNA specify the order of amino acids in a polypeptide. So again, amino acids, polypeptide. Now as we know that inside a protein, amino acids are present and polypeptides connect them. So DNA actually, based upon the sequence of the amino acids, you can decide the information which is to be contained in DNA. So DNA will specify in what order the amino acid should be present, because based on the sequence of amino acids, a particular characteristic will be determined. And RNA is used in the synthesis of polypeptides. That is, it is used in the synthesis of proteins, because only if you synthesize proteins, only then you'll have the sequence of amino acids. Only when you have the sequence of amino acids, you can put them in the right order to have the correct character. So that is where the nucleic acids play a role. So they are closely related to the proteins or the amino acids. So let us talk about the structure of nucleic acid. So if you talk about their structure, they are made up of repeating units, like how carbohydrates are made up of sugars, the monosaccharides, the way proteins are made up of amino acids. Similarly, nucleic acids are made up of repeating units called nucleotides. So several nucleotides get linked together to form a nucleic acid. So let us try to look at the structure of a nucleotide. So when you talk about a single nucleotide, it has three parts. The first part is a phosphate group. So it has a phosphate group, as you can see here. This is the phosphate group. The next part is a sugar. So this sugar can be either a ribose sugar or a deoxyribose sugar. So you remember while we were talking about monosaccharides, I told you most of the saccharides or carbohydrates, they end with OSE, like glucose, fructose, ribose. So this sugar can be either ribose or deoxyribose. So here this portion denotes the sugar. So this is phosphate, this is sugar, and the third part is a base, or rather a nitrogenous base. Now this base can again be in two forms. It exists in two forms. One is a single ring form, the other one is in the double ring form. So the single ring form bases, they are known as pyrimidine or pyrimidine, whatever you call it. So they are called pyrimidine or pyrimidine, and the double ring bases are called purines. So these are the three parts which make up a nucleotide. So here you can see these are the three parts. Now what are the various bases that we see? We will look at their examples. So when we talk about them in detail. Now the base changes when we talk about DNA and RNA. It is just the base which is different. Otherwise the phosphate group and the sugar part remain, even that changes. The sugar part also changes. So right now in this slide we will be talking about DNA, that is deoxyribonucleic acid. So for DNA you have a phosphate group. The sugar here in this case is deoxyribose. And what about the base? There are four bases which are possible to exist in the structure of DNA. These are the four bases: adenine, guanine, cytosine, and thymine. So they are abbreviated as A, G, C, and T. So just now I told you that nitrogenous bases can be of two types: single ring structure or double ring structure. So here by looking at these structures you can tell which all falls under the purine category and which all fall under the pyrimidine category. So adenine and guanine are double ring structures, so they are purines, whereas cytosine and thymine are pyrimidines. Right? So all these, now when you actually look at the structure of a DNA is a double stranded structure. That is, you have two strands like this. So you'll have two strands this way. Like you can see the animation here, there is one strand like this, there's another strand like this. And you can see some bonds in between the two strands this way. This way some bonds exist. So what is this bond? Now this arrangement on each side is of the base, and the bases form bond with one another. So how do they form that bond? The bond always exists. The adenine always pairs up with thymine and guanine always pairs up with cytosine. So that is how the pairing take place. So here in this picture, if you look at the structure, you can see all the green structures are adenine and all the pink structures are thymine. So green pairs with thymine, so green pairs with thymine, right? They always pair up together. Similarly, the blue structures and the red structures, that is guanine and cytosine, G and C, they always pair up together, as you can see here. And that is how the two strands are connected to each other. So the two strands, this is one strand, this is one strand, this is another strand. They are linked to each other by the bonding between the nitrogenous bases. And what is this backbone made up of? So this strand which I am talking about, that is made up of the phosphate deoxyribose. That is, the phosphate group and the sugar together form the backbone, and the base forms this bonds because of which the two strands exist together. Right? Now the speciality of DNA is that they have the capacity or ability to duplicate. So they can duplicate themselves, they can form a duplicate copy, and their function is to store the genetic information. Right? So here we can write that the phosphate deoxyribose forms the backbone. So this is about the structure of DNA. Now similarly, let us look at the structure of RNA, that is ribonucleic acid. So in this case, the only difference is that the phosphate group will remain the same. So this is the phosphate group. The sugar group is ribose sugar in this case. So it is ribose sugar. So what is the difference between ribose and deoxyribose sugar? So if you look at deoxyribose sugar, there is only one OH group. There was only one OH group. The other was H. But here in this case, there are two OH group. In case of ribose sugar, there are two OH group. Now due to the presence of two OH group, ribose is less stable when compared to deoxyribose. That's because when you have two OH groups so close to each other, these are more prone to hydrolysis. So you can cut them down by adding water. So that is why it is less stable when compared to deoxyribose. Now if you talk about the base here, now there is a slight difference here. Like in DNA, you had 4 types of bases, that is A, G, T, C. Now here adenine is there, guanine is there, cytosine is also there, but instead of thymine, you have uracil. Now, why don't you have thymine here and why do you have a new one, that is uracil here? That is because since there are two OH groups present now, so the thymine group, if you look at the structure of thymine, you will see that the thymine group getting connected here will make the entire compound very less stable. That is why it is replaced with this structure of uracil. So here how the pairing will take place between the bases, here adenine will pair up with uracil and guanine will pair up with cytosine, and this is a single-stranded structure. So RNA is a single-stranded structure. So what is the purpose of RNA? Its purpose is to take part in protein synthesis and also to convey the genetic information stored in DNA. So DNA will store the genetic information and RNA will convey the genetic information which is stored in DNA. Right, so this is pretty simple. So here also if you look at the backbone, so this is the backbone which is made up of the phosphate sugar, that is the phosphate ribose forms the backbone, and these nucleobases are nothing but the nitrogenous bases, and the backbone plus the nucleobases together will form the nucleotide. So let us have a quick comparison between DNA and RNA. So structure wise, this is single stranded whereas DNA is double stranded. Talking about the base pair, you have uracil in RNA whereas you have thymine in DNA. RNA cannot self-replicate, or whereas DNA can self-replicate themselves, that is DNA can be created by DNA, but RNA cannot be created by RNA. RNA has to be created by DNA again. So if you look at their structure, RNA is less stable due to the presence of two OH groups, whereas DNA is comparatively more stable because they have one OH and another H group. So these are some of the differences between the structures of DNA and RNA. So basically when you talk about their structure, the nitrogenous base plus the ribose sugar or the deoxyribose sugar, they together form the nucleoside. The nucleoside along with the phosphate group will form nucleotide, and many nucleotides together will form polynucleotide, and polynucleotide is nothing but nucleic acid. So this is the composition of nucleic acids. Thank you. Please visit www.examfear.com to watch more videos, attempt free online test, get free study material, find tutors and mentors. Thank you once again.